Good morning, everyone. My name is Bruno Trevisan. I'm a PhD student at the Federal University of Goiás, and it is my pleasure to welcome you to this session presented by Dr. Salvador Gezan. Salvador Gezan is a statistician and quantitative geneticist with over 20 years of experience, currently serving as a statistical genetics consultant. He specializes in mixed models and their application to biological problems, supporting agricultural, forestry, and aquaculture breeding programs, and evaluating phenotypic and genotypic data, defining optimal breeding strategies, and implementing genomic selection in operational breeding programs. He began his career at Tottenham Research and was an associate professor at the University of Florida, and has delivered SVREMO workshops globally. Dr. Gezan has over 160 peer-reviewed publications and co-authored statistical methods in biology, design and analysis of experiments and regression. Before we begin, I'd like to remind everyone that we'll have a Q&A session at the end of the lecture. Please feel free to submit your questions and comments in the chat throughout the presentation. Dr. Gezan, it's a true privilege to have you with us today. On behalf of the organizing committee and our audience from around the world, thank you very much for sharing your time and expertise with us. You may begin.
Hello, well, good morning, good afternoon, or good night. Depends where you are. I'm happy to join today. So please bear in mind, I don't have a very good internet connection, so if it gets a bit sloppy, let me know. But hopefully all runs well. So I want to talk about some of the more complex variance covariance models that we use for G by E. And this is a very large topic, and I didn't want to talk too much into the statistical details. I want to present a little bit more about some of the issues and how we face this problem. So we use a bit more complicated models that will give us a slightly different insight. I also will bring some things from my own experience from consulting and some work in different manuscripts and students and etc. companies. So elements to consider when you move into these types of analysis, okay? So can you see my screen there? All fine, as I move the slides? They're good? Years and even sometimes different types of management. All of those, like in a generic way, we call them environment or we call them location in a generic way. So why do we want to do MET? We have different reasons, and you probably heard a lot of this about in the previous talks. So obviously we want to estimate total genetic values or breeding values for the genotypes that are being evaluated. And because there's a bit of an imbalance, obviously we have a complication in there that genotypes are not in all the places, that they're not sampling all the conditions. Also, we estimate variance components associated to it, and some of the variance components and the calculations associated with the variance components, such as heritabilities and type B correlation, are super important for us to decide where is the level of G by E and where is the conflicts are happening. So it is important for us to know how much G by E at the end we have. And then understanding a little bit with a bit of the more complicated models, we get also ideas of the potential structure across the site, across the environments, or across the different managements of how this G by E is being expressed in our population, and that helps us define our breeding strategies. So that's another reason why we want to look at these magnitudes and so on. And obviously, at the end of the day, what we do is we want to select individuals. And we want to select individuals in some cases that are very generalistic, so good across these environments, or individuals that are more specific for specific markets, environments, conditions, etc. Okay. So We get data sets, and the data sets for these are usually very messy, unbalanced. And there's an example on the site where we have 10 environments. Not all the genotypes are in all the environments. And this creates obviously complications. Not all the environments have the same number of genotypes. Sometimes they are completely disconnected. Sometimes there's individuals that are more in common. And sometimes you have an individual only in one of the environments and nowhere else. And remember, what our goal in general is to get an estimation of the performance of the genotype or across the environments, which goes along the columns, or we want some specific values for the cells, for which some of the cases we have no information. One of the things that I always emphasize with this is that we need to make sure we have good connectivity, which means obviously then we need to have some common genotypes across environments. And the best way to think about this one is that at some point you're going to estimate the correlation on the rankings between site 3 and 5. And in order to compare site 3 and 5, you need to have a decent number of genotypes that are in common between these two environments to estimate that correlation. So that's the sort of connectivity we focus on. Obviously, we have lots of plots, lots of different types of experimental designs. Sometimes variables are slightly very differently, not completely standardized how they're defined. So we have other sources of noise, which is good to kind of manage on this. So this something goes with the protocols, different types of plots and so on. And all that kind of translates into lower heritabilities, but sometimes some of these things translates into increased G by E that are not due to the E, are mostly due to some other issues related to how things were measured, okay? And then obviously we need a good design for all of this. So there's plenty of software that generate nice single site designs like alpha lattice or row column designs, or something that is a bit more complex like the sparse designs and so on, okay? So hopefully you can still hear me. I'm going to talk about the basic model. This is one of the historic models that we obviously see everywhere. We have an overall mean, we have an effect of the site or the location, whichever name you want to give it, and we have the interaction genotype by environment or genotype by location, and so on, and the error. Now, this is the traditional model, and this model is the one I learned when I started doing my PhD, and even probably on the undergrad. And it's the base model is called an interaction model because the G by E is modeled as an interaction because we have the main effect of genotype, and sorry, I forgot the G there. We have a main effect of genotype, and we have the main effect of site, so the GS is an interaction. So this model, you can see here in a bit of a notation. There's a few elements that I like to always consider when we try to fit this MET model. Obviously, we are going to have different fixed effects associated with each of the sites. For example, replicates is different. We have the main effect of the site or environment, which could be random too, and there is value on having them random in some of the conditions. Then we have some random effects associated with design, for example, incomplete block designs associated with each of the sites. And then the main effect of genotype, which is going to give me, at the end of the analysis, a blood value that is the performance of each of the individuals across the whole experiment. And then we have the G by E. Now, this G by E might be our main focus, but also you can think about this G by E as a noise. It's the inconsistency of the genotype, that it is not always the same genotype across all the sites. So those deviations, some people like to see this one as a source of variability, more like a different type of error or residual. And then obviously we have the typical error residual that we manage. Now there's one thing that I have defined since here, but I think one of the things I want to emphasize is this model always fit. You have tons of software, historical software that fit them. But one of the things that is important to consider, it's a flexibility on the model, and the flexibility that I almost never forget to add is the heterogeneity of the variances of the errors. So that means each site has a different error variance. So they are heteroscedastic in that sense. And this is very important because each of the sites have possibly different scales, different levels of heritability, different sources of variability, and so on. Consider a common error is almost like saying they are all measured with the same precision, which is not really the case. So that's a very important element to consider. So the same model where we fit it in AS-REML. I'm just presenting AS-REML. We have the main effect of site, we have the main effect of genotype, and then we have the interaction site by genotype. And here I'm defining heterogeneous errors on the residual part of the R matrix. Nothing new on this. This model always fits. It's a really nice model. Computationally, it doesn't take much energy because you can have hundreds of sites, even hundreds of thousands of genotypes with or without pedigree or genomic matrices, and this model is going to converge nicely. But it has restrictions. So first, one of the restrictions is that it provides a single correlation across the environment. More than provide, assumes that the correlation between any pairs of sites is the same. And this is associated with this variance that is the same that applies to all the sites. And so that's not necessarily a bad thing, but it is a bit problematic. And the other thing is assumes the magnitude of the genetic variance is the same also on all the sites. There is different error variances, so there's different heritabilities, but like the signal of the genotype is the same. Now, this could be a very good model if you know your genotype, if you know that your environments are more or less similar, that your effect of the genetic signal is more or less the same, your scales on everything is more or less the same, and so on. And it has been successful, but it is very restrictive. And I almost would say this is kind of like an outdated model, okay? Because that's not really the case. I've been in cases where the variance, the phenotypic variance, just a phenotypic variance on a group of sites, it goes from 2 to 280. So, and because of that, you have a whole array of magnitudes of things, okay? So now we go to the modern model, which I like to call it more like the model where we should be using. And that's like a 20th century model. So this one has the same fixed effects, also the sites as fixed effect, the design effects as random effect, but it has a single term of G by S, which means it's a term that is the nested effect of the site within, of the genotype within site. So this is where you will get the blood values of this model, where each genotype has an effect on each of the combinations of sites. So it's a nested effect of genotype within site 1 and genotype within site 2 and so on. And this means there is a bit more complicated because that matrix of all those effects is separated into a Kronecker product where we assume the genotypes are independent, but we can change that to a GRM, or we can change that to a pedigree matrix. And also we have the G matrix associated with the variance covariance of the different sites. And this is where we add different things. The rest is more or less the same. By the way, you can also get your effect across all the sites by just averaging your prediction of the genotype 1, 2, 3, 1 inside 1, and 1 inside 2, inside 3, and so on, and you just collapse them, and then you get that. Okay, we fit this in AS-REML as without the genetic effect, and then we have just core GH, which is one of the most complicated structures, which I'm going to define. And this is where I really wanted to mention, these are the complicated models we fit. Now, particularly the core GH or the US, which is the ANI structure, where you have a different variance and a different covariance for each combination of sites, it gives me a type B correlation that is different by pairs of sites. So it allows each site to express that. And then also gives you a different variance for each of the sites. So the diagonal elements are also different. And it gives you a lot more insight, and it weights different the information from those. So the typical structures of G are IDV, which everything is independent. Imagine you have 4 sites here. Yes, that's okay. It's actually an incorrect assumption because they are not independent, because we assume that the genetic effects of the individual in site 1 and site 2 are correlated. The diagonal is similar, it just allows me for heterogeneity of variances. So again, it's also an incorrect model because we know we have some covariances or some correlations between pairs of sites. Their genotype, effect of genotype in site 1 and site 2 are correlated. And now we come up to the more complicated ones. Core V is a nice one where we assume the correlation on all of the elements is the same. That can be expressed as a compound symmetry structure, or is actually equivalent to the interaction model that I expressed before. The interaction model is defined by a variance by a genotype genotype by site plus a genotype. And this one is being defined by a correlation and a variance. So they are different parameterizations, but they're exactly the same model. Now I'll show you that. This has this strong assumption that everything has the same variance. Better than that will be to have the same correlation, but different variances. And this again assumes that the correlation is estimated across all of them. And this is a very nice one. It's one of my favorite. And then you have the US and the core GH, where you have different correlations between pairs of sites. This is really the holy grail. This is where you want to get to. You want different variances per site and different correlations. However, sometimes you cannot estimate things. For example, there is no common genotypes between site 1 and 4, so there's no information. Or sometimes you have lots of parameters to estimate if you have too many sites and the system cannot converge. So I'm going to work in an example. Hopefully you can still hear me. If somebody says yes, that'll be nice. So we have the weak data set here. This is an example that we had pulled from a paper, and it's in the library ASR Triala. And we have used the ASR Triala's functions to fit this model, and everything is using ASRML in the background just to illustrate things. So we have the response variable, which is the yield. We have some sites with 2 replications, sites with 1 replication. Everything is alpha lattice, and we have 3 checks and 270 lines. The majority of the lines are in all the places, except for a few little bit mortality. So everything is very good in this case. There was a 2-stage analysis done on this. On the first stage, you fit every model individually, and then you get the predictions, which are the blues. Some people call them blues. I should call them predicted blues. And the weights that are being associated with those. So we move into a 2nd-stage analysis. But this can also do in a single-stage analysis. But you can see here the way the data looks. There's a mean estimated for each of the genotypes. And then we have the standard error associated with that prediction. And we have weights. And you can see the weight is much larger on the first 3, which are actually the checks that have a lot more replications than the other lines that have 2 or 1 replications, okay? And so on. There's just a couple of sites in here. So I don't want to go too much into 2-stage analysis, but it's one approach in which you analyze first each of the individual sites individually with the spatial components, covariate, look for outliers, and so on. And then you get a blue prediction, blue mean, and the standard errors. And also you can get something that it's a type of weight, which is the inverse of the diagonal of the inverse of the variance covariance matrix, which is, there's a few papers that support to use that one. On the 2nd step, you collect all that information and do what is known as a weighted analysis. Pretty much it's a meta-analysis. And that's where we incorporate the same structures. The results are almost identical. So you shouldn't worry about that. Everything I'm talking here will apply also for the single-step analysis. So let's go back to some of these things. Here you just have a few of the codes of how they were fitted, some of the things, some of the different elements. Actually, these are the same, but this is the core GH. This is the interaction model, has only 2 variance components. The core V has also 2 variance components. Remember, the interaction model has the main effects of genotype and genotype by location. And the core V has main effect of the G by E. And notice this 95 is the sum of these 2. Exactly the sum of these 2 as it should be, because things are being nested. And it has this correlation, which is the type B correlation estimated the core V. I can also calculate a type B correlation based on the interaction model. And if you do this 45 divided by the sum of these 2, you get to exactly the same value. The log likelihoods are identical. Sometimes you see small differences. And AIC and BIC are identical. There'll be small differences. The diagonal is clearly an incorrect model. Somehow it looks a little bit better in this particular case. It has different variances, but it's ignoring the correlations. These 2 models have implicitly, this one a correlation, and this one is explicitly as a correlation. And then we have the core H, which is heterogeneous error with the correlation. And you can see the big improvement that goes from 8 to 9, just an additional variance component. And this one, it's a little bit of a gain, not considerably, but there's a little bit of a gain. And here I have the core GH, which is the model where we have, remember, we have 8 sites. So we have a matrix of 8 by 8, and we're estimating every single variance and every single covariance. I put this one as reference, and we are lucky to fit it, but when you have too many sites, you cannot fit this one. So what's happening here is, obviously this is a much better model. You can see a big jump of this, but I'll show you a few things as we come up with this one, okay? Because not always you get to this model. Look at here, you see the diagonal elements, so it's a different error variances. They are not very different in this particular case, particularly when you see the core age. You have a very similar correlation between environments. This is not very high and it's not very low, but it still is positive, but that means there is some G by E on this in this experiment. But we don't know if it's maybe one environment or a few genotypes. We don't know the dynamics. What is important to see, these are all genetic variances, and you see they range from pretty much 64 to 155. So there is quite a doubling, but not as extreme as I've seen in other ones. But it's still justifying that you have heterogeneous errors because something is going on that is different on the sites, okay? And so on. So I haven't associated the standard errors, but it's important to see the standard errors, okay? Now, as I mentioned, my goal is to try to get to this core GH, which is, in this case, will be 8 by 8, but sometimes we analyze things that are, and I'm not kidding, 120 by 120. So we cannot do anything with that. Often when you have more than 5 sites, particularly when not everything is present on everywhere, which in this case, the majority of the genotypes are present everywhere, but when you have all the levels of imbalance, more than 5 sites, you will have a really difficult time to fit the model. So we fit an approximation, which is a factor analytic, and I'm sure some of you have heard it. And you have we have to consider the factor analytic as an approximation to this core GH that we sometimes don't get to it, okay? So sometimes, even in any of these models, we have problems in fitting the models, okay? Particularly if you're trying with a core GH or a core H. The main problem is the connectivity, okay? So there is issues on the connectivity. There's no information between pairs of sites. You can't estimate a correlation, or the correlation could be -1 on 1 because you have only a few points on that one, okay? So there is nothing in common. Sometimes there is nothing that you can estimate. Sometimes you have things in common, but they are not very good. Scales, very different scales also create issues because see, the variances are very different. And sometimes the way to get around this one is to use some standardization, like dividing by the phenotypic variance and so on. And some crops use that. Too many variance components to estimate. You need to provide a starting value. Sometimes everything is fine, but there's just too many things to estimate. And that's one of the reasons why often we tend to use the 2-stage analysis. So the variance components that need to be estimated are only associated with the G by E. Outliers are always a problem, and they need to be eliminated, ideally individually by site. The sites that you pick in order to estimate some of these elements have to have a non-zero heritability. A heritability of 0 on one of the elements on one of these variances, it actually creates a problem. So it doesn't allow me to estimate, for example, some of the type B correlations, and the whole system becomes very unstable. So eliminating sites that have low heritability or no heritability is important. And then also doing a very clear selection of the sites, sometimes because of the scales or for issues, it tends to help, okay? And also don't try to fit the most complicated model if it's really not what you are needing. So that's why some other models like core H is very handy, or even the core V in some of the cases. But we can go into the factor analytic as a way to approximate this G of 8 by 8, okay? So this G of 8 by 8, sorry for all the noises, it can be approximated with a factor analytic. And the factor analytic, you can see it in 2 ways, as a way to approximate the G matrix, and you don't care what happens inside. It's just an approximation that is based on fewer elements, and it might have some biological interpretation or not. Or you see it as a way that it fits this G by E, but in addition, it gives you some good biological interpretation or the possibility to extract some other elements, particularly associated with the loadings that provide information about the underlying environmental factors that are inducing this G by E. So there is some theory about it. There's quite a good number of papers, and they use this in many different ways. So I do recommend, if you want to take a look at it, have a look, okay? So how do we do this? We fit this, for example, 4x4. This is what we want to aim to, but we're going to fit this with loadings. And we fit this with loading, which are this one-factor loading we call FA1, and the transpose of the loading, and some specific variances that are these. So these are kind of like the explained portion based on an underlying loading, and these are the unexplained portion of the G/E that we cannot explain by these loadings. You can increase that to of FA2, and that allows you to have 2 loadings, and you can see now here I need to estimate a variance component plus another 4 variance components. That's 12, which is actually 12 is more than the 10 I need to describe this process. So some of these variance components are going to be fixed to 0. You don't have to worry about the details, but that's really what is happening internally. Okay, let's go back to our example. What I did here is I fit FA1 to FA4. So I expanded the previous one, and I have the core GH, which is the maximum 36. FA4 actually is overkill. It is more than 36 elements, but some of these variance components were set to 0. So anything that is kind of fixed to 0 value, because it explains some of the specific variances to 0, is not counted. But you can see here that we increased from 16 variance components, 22, 29, and then we go to 33 or 36. You can see the progress, and there's a big jump in FA1. Actually, even from any of the other one, there's a big jump, and there's another big jump from FA1 to FA2, and then there are marginal jumps, and some of these significant, some of them not. And this is what I have here. I have a significance that I compare the FA2 against the FA1, 6 degrees of freedom. This is a likelihood ratio test. This is highly significant. Then FA3 compared to FA2 is highly significant. And FA4 compared to FA3 is significant at the 5%, not at the 1%, and so on. And this is the comparison between core GH and FA4. And you can see there's no significant differences because they are pretty much describing the same thing. Okay. On top of that, you see the correlations between the breeding values, in this case, the genetic effects or the predictions as random effects, and they are all very high. Obviously, the diagonal is really poor but in this case, still very good. But it's relatively a little bit more poor than the other because that's an incorrect model. But you can see almost all of the models give you very good things. Pretty much the take home here is that anything like an FA1 is already so close to the core GH, maybe not statistically to give you all the insight on the G/E, but in terms of providing you with the predictions that are very close to what you will get when you analyze everything, it is very, very good. And even the interaction model is good. Now that is going to depend on the type of data you're analyzing. It's going to depend on the dynamics, the levels of G/E. If you have negative correlations between environments, all of these are positive, then that creates a more instability, and different models might kind of be more erratic. Okay, but this is a very, very good result. Now, which model will I pick here? I would probably pick FA4 if I use AIC. BIC is more conservative. It's a good model, but you have to also keep in mind, what do you really want? I want to have a better insight of the correlations, even if they are not much differences and maybe estimated a bit poorly. So I will go with FA4 if that's my goal. If not, you can stay with FA2 for your predictions and you won't have any more differences. In this particular case, I'm going to focus on FA4, okay? So now let's go back to the variance components. I added the other columns for FA4, core GH, and we have core H. And you can see here the variances, in this case, FA4 with the core GH are almost the same. There's a few differences here. They converge slightly different, but there's really not much to say that they are effectively different. And you can also look at some of the correlations. This is the correlation between side 1 and 2, 2 and 4. And you see a few big differences, 0.798 and 4.62. Now, what is not reported here is the magnitude of the standard errors, which are probably very large. And this is because we have only one replication in the majority of the sites, except on site 1 that has 2. All the other sites have 1 replication. So there is possibly a lot of uncertainty on the estimation of these correlations. So you see a little bit of differences, some more and some less, but I suspect the standard errors are around 0.2. So what these correlations tell me? They tell me lots of things, and we can do lots of things with this matrix of correlations that we'll obtain. They obviously allow me to screen the sites, to see if there's a site that is an outlier, to understand what are the dynamics of G by E. Maybe I have 2 main clusters. Also, the correlations, when they are high, they are telling you that you are borrowing more information from the one side to the other side. And obviously, when the correlation is 1, is you are borrowing a lot of information. When the correlation is 0, you're borrowing non-information. But it's very, very interesting. So this is called effective degrees of freedom. So you borrow more information without correlation, and allows you to even eventually eliminate some sites. A heat map will provide you a different insight. Now, all of this, you have to use it with care. I mentioned already that some of the standard of these correlations are very big. If you have poor replication, you expect that to be not very well estimated. It still gives you insight. So again, when you have single site analysis, unreplicated trials, you have to take it with a bit more pinch of salt. But also make sure that you are really on your objective to fit the model. So if you really want to fit a model that is complicated to get more insight, then that's the case. You need more correlations. But if you really only want to have a generic model, for example, a core H, then you only estimate 1 correlation, and that's probably going to estimate with reasonable precision and so on, as we had with this 0.475 or 72. Okay, now here a bit more insight on this. So we have a heat map on this, and you can see one side, which is clay center, that is a bit off from the rest. And this means maybe this site has problems, measurement problems, but maybe that is a different soil type. Then you have this really nice cluster here that is representing something. You might associate that with a market area. On the side, I have a biplot where you can see the lines from the different environments. The closer the lines they are, the higher the correlation between the sites. And you have the dots, which are the specific lines. You can see here one of the check, which is Freeman, which is really far to the left. And then there's the site, it's aligned with some of these lines. That means it's a very good response or prediction on the value of that one on those environments. And in some other ones, it's not as good. And again, that gives you a little bit of other insight that you can use on this. So these lines are associated with the variance, covariance, and the correlations. Again, here is the matrix of correlations between every single part based on an FA4. You have this value 0.16 as the lowest correlation, and the highest is 0.857. If you take this average, you'll be around in 0.45, which is what the core H is giving you. Now, there's a few more insights on some of the results. These are comparing the different models and how much the genetic component it's explaining, particularly the loadings. How much the loadings are explaining on an FA1, which has only 1 loading with respect to the full G by E. This one is how much 2 loadings are expressing, 3 loadings, and 4 loadings. And you can see when you calculate the difference between these two, you can see that there's diminishing return on how much the additional loadings are really explaining. By the way, sometimes this one goes up and down, this column goes up and down, because instability on the estimation of the components. So this is an idea to tell you what are the underlines for the specific FA model, what are the magnitude of the underlying effects, how much they are influencing the G by E. And we know there's a lot of more influence in the component 1, which we don't know what it is, but it could be related to temperature, precipitation, distance from the sea, and so on. This is a nice one too. It's based particularly on the FA4. It tells you how much the loadings are explaining with respect to the total genetic variance by site. So there's 2 sites here, Grandee and Lincoln, that 100% of their G by E has been expressed by these 4 loadings. And this is the one that have a specific variance of 0. And there's one side here, you have Clay Center, and another one here that have 50% only is being explained by the loading. That means there's a big portion that we don't know what it is, and it could be related to environmental errors and so on, etc. Okay? Now we get our predictions. We can get our means predictions across for each genotype. It's good to add a few things, if they are being extrapolated or not. Sometimes you want the predictions on combination location by genotypes and want your predictions of a genotype across all the locations. And here, as you can play with this and take the average of all the sites of, in this case, Freeman, across all the sites in different ways. Maybe you have defined in 2 zones, maybe you want to leave Clay Center out, and we only average these 4 environments, which are Alliance, Grant, and Lincoln. So we average only those predictions, Alliance, Grant, and the 2 Lincoln, as one ranking that we want to try to see if we commercialize something, etc. So there's many different ways that you can use this. You can use weights to define those averages by group, by breeding zone, etc. Okay, and obviously you can redefine it with other criteria like markets and so on. I like, and I suggest that you add other elements. We can add things like reliabilities or standard errors or some measure of the precision, but also how many plots or how many sites this genotype was evaluated, or how many plots, and so on. And that information helps the breeder to make their decisions of what to pick. We can do a stability, and stability is when we want to see if we can select individuals that are stable across all the environments. We have different measures. I'll use one called static, but there's many more. And also the FA analysis allows you to use their loading as a measure of stability, and there's different papers that help you to do those things. This is important. Sometimes we don't care about it because we're selecting the genotypes across everything, but sometimes allows us to get some insight and make slightly different decisions. And remember, there's a lot of graph like spaghetti plots and other more that help you also to get the insight of these predictions and try to make a better decision. So when we use the factor analytic, we can use these loadings that are associated with each of the genotype, which is almost a slope associated with how sensitive is a genotype when we change the environmental covariable that is underlying. Okay, so imagine there is something like humidity that increases as you go closer to the sea, and that produces an increased response on the mean, or a decrease, but let's assume an increased response on the mean. And obviously, the more steep that slope is, the more it's going to be reflected as a G by E. Okay, and then we have obviously used those things together with some plots to see if we select individuals for each of those conditions. Okay, and you can expand this to do environmental variables, and there's a few papers that do that. So the 2 things I want to show here, and I still have a few minutes. This is the predicted value. So this is the performance across all the sites. You can see the performance of the genotypes across all the sites. The higher the performance, the better the genotype is. And here you have Freeman and some very interesting ones. These are very poor ones. But the static means that the lower the value it is, the more stable the genotype is across the different environments. So here you have the Freeman is good, but it's not as stable. And you have this NE16402 that is very good, not as good as Freeman, but it's a lot more stable. So again, you can define things. This is looking at the loadings, and I'm just going to show 2 genotypes here, mostly for the first factor, which is the first loading. And you can see the loading increases the loading, and that means there is a slope associated with it compared to this one where it's almost flat. And if you have this slope associated, there's a group of, for that individual, and there's a group on the locations where this individual actually has a higher performance compared to the left side. And you might eventually say, oh, I'm going to pick this individual for this environment, but not for these conditions. Notice that the mean here are all very large. They're +10 to +20, compared to this one where the breeding values are almost 0. And you have this location, so this genotype on this location seems to have a noise. Now remember, there's only one observation in the majority of the sites, so this could also be an outlier on the performance of that individual. Okay, but there is good things to do this. You can explore, you can do different things, and this is again to feed the breeder. Okay, so just to close a little bit, and obviously there's many more things to do. I just show you that we can fit more complicated models, and we can use a factor analytic to get a bit more insight. But it's very important that when you do your METs, you align your analysis to what you really want to obtain, okay? So we have the estimation, which is really estimate the magnitude of the G by E, the variance components, the type B correlations, and again, precision related to the heritability associated or heritability without G by E. Prediction, we want to, in some of the cases, and this is prediction on the sense that we can expand this model with some environmental covariables, something I didn't present here, but you can use some of the factor analytics too, to predict what will be the performance of some genotypes and untested environments, which is the enviromics. Very popular these days. And there's a few other things where you want to predict also, and this goes with the genomic selection, which is predicting the performance on individuals where they haven't been tested, if we consider a genomic matrix and so on. Deployment is to decide what we are going to grow commercially, where we're going to establish it, what do we expect to have in performance, because we have analyzed these trials. There is also selection. What are we going to select when we deal with breeding values to be the parents on the next generation, on where are we expecting to test the offspring of those parents that we're going to select. And also to simplify, knowing the level of G by E that you have in your breeding program might allow you to simplify from having 4 breeding zones to having 2 breeding zones, or in some cases just 1, which simplifies some of the things. Maybe not all the traits act the same way, but some of the traits act the same, are very handy. So do we really need to fit complex G by E models? Well, depends on what you want from above. I do expect that complex models will improve our estimation of many things, but even when you fit complex G by E models, your G improves because your G by E is better estimated. So even if you don't care what happens with the G by E, you will gain something on the G with slightly better models. Some sites are outliers, and understanding those correlations when they're really poor, then it gives you a little bit of an insight that something is going on, or there's another market, or something that you might need to do. Do you have the insight? This goes with the insights again by the breeding zones and so on. And finally, one thing that is very important is you're only going to be able to estimate this correlation and these variances if your experiments are reasonably good, which means that they're well connected and there's a reasonable number of genotypes that are common between sites. It doesn't have to be exactly the same. So, but there's a few elements that make these models a bit suspicious or unstable. And in that case, you need to be a bit careful. Okay, so with that, I finish. Probably a good time to allow for some questions. You can always contact me, and I have to thank VSN to allow me to spend the time preparing this presentation. Hello? Hello.
Thank you very much, Doctor. It was a pleasure and an honor listening to your presentation. We'll now move on to our Q&A session. Camila asks, from a practical breeding perspective, how much can spatial analysis change genotype ranking or selection decisions? And what diagnostics do you consider essential before trusting spatially adjusted genetic values?
So that's a good question. If you're using unreplicated trials with poor replication, I feel spatial analysis is critical to perform to eliminate some of the bias. So for me, it will be a must do on unreplicated trials. Now, when you have more replications, if your experiment is good, so you have, let's say, 6 replications and you have an incomplete block design, you will almost no need any spatial analysis, and simulations have proven that. But you can always gain a little bit. It always gives you a little extra. And if you can implement it and it's computationally not too hard, then it's a win. It's like a low-hanging fruit. Okay. More questions?
We have a question from our previous talker, John Zanon. He says, hi, Salvador, big fan here. I literally watch all your videos. I have 2 questions. I'm going to give you the first question and you answer, and then I'll...
Yeah, no problem.
So his first question is, in your experience, what usually limits prediction accuracy the most? Data quality, experimental design or model choice?
Model choice, not so much. What was the other one? Data quality and, sorry?
Experimental design.
Experimental design. My experience, particularly with METs, it's the data quality. It is very common sometimes when some breeding programs go across different countries where there's no much of the standardizations of the protocols, how things are measured or how things are established. So think, for example, if you have a scale from 0 to 10 for some disease, there's no very standard. Or things when they have to measure, the plots are of different sizes, or the managements are very different. So you need, in order to analyze all this properly, you need to have very well standard protocols. Otherwise, everything ends up being translated into GBA. So models, you can play with the models. Design can help you if you have decent replication. It's less of a concern. But for me, it's protocols.
His other question is, when designing a multi-environmental trial network, what should be prioritized? The number of environments, genetic connectivity, or environmental diversity? Is one more important than the other?
So, again, it's going to be very personal. It's going to depend on the program. The trend has been that we want to favor as many environments as we can, particularly, well, in many stages with the unreplicated trials. So the idea, instead of having, I don't know, the way I see it is instead of having 5 experiments with 5 replications, each of them, rather to have 25 experiments with 1 replication in each of them. And that's been the trend. Smaller experiments, logistically a bit more difficult to manage. But in one sense, what you're doing is your genotype is sampling more conditions, different environments. Now, again, 1 replication, I consider it a bit poor, especially if what you want to do is have some insight on the ranking within a location, which sometimes is important. So maybe you have to come up with somewhere in between where you have 2 or 3 replications. So hopefully I answered the question. So, yeah.
Thank you. We have one more question from Mateus Chagas. Hi, Salvador, very good. What additional metrics beyond EVV or TGV accuracy should be considered when assessing the quality and robustness of our predictions across different environments?
Across different environments. So one of the things I didn't mention, and I'm a big defender of reliability, is remember actually when I was presenting, there are some genotypes that were never present on some of the sites. And they would have a prediction because there's correlations of the genetic effect with the other sites. But when you calculate the ranking of that of the genotypes on that given site, knowing that some individuals were never present, it might give you an additional information. And sometimes you group 4 sites and another 4 sites, and one genotype was never present in one set of the 4 sites. So it is important because you can decide if you add the number of sites or you add the reliability will tell you how much information you have on that individual, kind of like extrapolating. There is something that also you need to consider the other way around. A genotype might be in a bad, relatively moderate ranking on a site, but it has a very good ranking on the other side. And because it's not present, it's been shrink a lot. So it's very close to the mean, but if you give it the chance to test it about, it's probably going to be much higher. So sometimes you end up penalizing individuals for some sites because they are not there and you think, no, this is not good, but in reality it hasn't been evaluated much there. So considering those, animal breeders use reliabilities, we could use something like reliability, but you can create even just counts of number of plots, the number of sites, things like that. Okay, hopefully that helps.
Thank you so much, Dr. Salvador Gezan. It has been a real pleasure having you with us today. We truly appreciate you taking the time to share your knowledge and experience with us, and you're more than welcome to join our next lecture.
I will do so. And again, if pleasure to be here. If anybody wants to send me a message, an email, and ask something, I'm totally open and happy to receive your questions too. Okay.
Thank you very much.
Thank you for the
Our next lecture will begin in just a few minutes, so we kindly ask everyone to move over to the next YouTube stream using the link available in the chat. We look forward to seeing you there. Thank you and see you in a few minutes.